For session number 1, we have 3 outlines. I'm going to be talking to you about quality control of your raw data, give you a background on Data2 pipeline, and presenting the first part of Data2 workflow for sequence RNA analysis. Before analyzing generated sequence to draw biological conclusions, a quality control check should be performed to make sure there is no biases in the data. QC gives a quick impression of whether your data has any problems of which you should be aware before doing any analysis. We do the quality control to see what kind of problems may underlie in your data. It's good to know which problems you do have to be able to do some corrections like trimming and filtering. The potential problems that can be found in your data are, for instance, low confidence bases. In this case, the sequencing instrument did not know whether it should be an A, T, C, or G base in the location, so it just puts an A instead. Another problem there can be is sequence-specific bias, or there might be also some sequencing contamination or adapters left. Normally, those adapters are usually removed by the sequencing facility. There are many software packages available for quality control. Here I have listed some of them. We have FastQC, which I think it's one of the most used. It's very nice and has a good graphical reports. We have also MultiQC, FastX Toolkit, Bransec, Tag Cleaner, and NGS-QC Toolkit. The raw sequence that you get from next-generation sequencing platforms are on FastQ format. FastQ is equal to FASTA plus quality. It's a text-based format for storing both biological sequence and its corresponding quality scores. In your FastQ files, you will have 4 lines per each read. Line number 1 begins with an @ character and is followed by a sequence identifier and an optional description, like a FASTA title line. Line 2 is the raw sequence letters. It's A, T, C, and G. Line 3 begins with a plus character, and it's optionally followed by the same sequence identifier. Line 4 encodes the quality values for the sequence in line 2 and must contains the same number of samples as letter in the sequence. So here you have an example of how a FastQ file containing a single sequence might look like. So the first line, so we have the @ character followed by the read name. In the second line, we have the actual sequence. On the third line, we have the plus and the name of the read, or some of other information regarding the read, or it might be also empty. And then the fourth line contains the ASCII characters, which represent the quality of each base. So there are as many characters here as there are bases here. The character exclamation represents the lowest quality, while the tilde is the highest. So for quality measurements, we use Phred score. So you can see here the formula. So if you have a probability that the base is wrong, then the probability for that is 0.01. If you calculate this formula, the Phred score you will find is 20. So 20 means that the probability that this base is wrong is 0.01. Here you can see an example. For the first base, T, the quality is 40, and so on. However, in the FastQ files, we have ASCII characters that represent the quality score instead of having those numbers. Maybe to save more space because there are millions of reads on your FastQ files. In this slide, it shows how the quality score is encoded. In the beginning, they have started with Phred 33. So here, from the exclamation mark, and make it as the trade quality score of 0. Then during time, new quality measurements were introduced, but now they are back to the Phred 33. So the FastQC tool, for instance, will give you which encoding was used. Usually you see FRUD 33 or 64. It's very important to know the encoding that was used to measure the quality if you would like, for instance, to compare different datasets. So what is FastQC? FastQC aims to provide a QC report which can spot problems which originate either in the sequencer or in the library material. The FastQC gives a very nice graphical report. It gives a summary of the modules which were run and a quick evaluation of whether the results of the modules seem entirely normal, which is represented by a green tick, slightly abnormal by an orange triangle, and very unusual by a red cross. The first summary consists on giving basic information on your FastQ files, like the name, the type of your files, the encoding that has been used, the total number of your sequences, and their lengths. So the next module of FastQC is the position-based quality plot. It's one of the most important modules in the report. This is the plot that you look at first when you get your data. Here is in x-axis you have the position, so it's going from 1 up to 36. So this is a 36 base long, and in y-axis you have the FRUD quality score. It goes from 0 to 34. And you have this nice coloring. As I said earlier, green is very good, orange is okay, and red is bad. The blue line that you can see here is the mean quality, and the red bar, those red bars, are the median value. So in this slide, I would like just to give you some examples how the good and the bad data will look like. In the plots on your right, the quality is second now towards the end. That is not good. Here maybe the library preparation or the samples were not good from the beginning. So FastQC gives you several plots. So you have also the per sequence quality score plot. The distribution of average read quality should be fairly tight in the upper range of the plot, as you can see in the plot on your left. Here the average quality is 37. You have also the pair-based content sequence plot. It shows the percentage of the 4 bases per position in the read. The proportion of each of the 4 bases should remain relatively constant over the length of the read, with percentage of A should be equal to percentage of T, and percentage of G should be equal to percentage of C. But for RNA, there is a clear non-uniform distribution of bases. For the first 10 to 15 nucleotides, this is normal and expected, and depending on the type of library kit used. So let's move to the next video.